Hello everyone, this is going to be my second video on how to get through my packets for genetics. This is the non-Mendelian genetics packet. So this is after the Mendelian genetics packet, after the Mendelian genetics packet, this is the non-Mendelians. This is starting with incomplete dominance. This is going to be a little bit different than stuff you did with your Mendelian packet. There's a little bit of differences between the phenotypes here that you didn't see in the Mendelian packet. So with all that getting said, let's show you what we're talking about. So in incomplete dominance, remember that you're the heterozygous form. You can see here the big B and the big W here. They're going to create a different type or a different phenotype for the offspring. So black fowls are big B or capital C big B, capital C big B. White fowls are capital C big W, capital C big W, and then the bluish gray or the grayish color, the in between, is going to be the capital C big B, capital C W. So you can see here, you have to write those down here just to remind yourself. And then it says to cross a homozygous blackbird with a homozygous white bird. So you can see here, we're crossing one of these with one of these. And you put the alleles, remember, up here on the side. So like you have a C big B, C big B, C big W, C big W. And they're all going to be that bluish grayish color. That's because they're C big B, C big W. So they're all that mixed, or the heterozygous. So the offspring are all going to be heterozygous, and they're going to be that gray color because these don't really follow Mendel's rules. They follow some of Mendel's rules, but not all of them. And you have to do this for all of these questions. You can see here each one gives you a different scenario. Moving on to codominance. Codominance is when you're going to see both phenotypes in the heterozygote. So we talk about roan cows here, and the roan cows that we're talking about are a mix of the heterozygous. So red cows in these cattle are in this case, I think the red ones are big R, big R. So this is going to be big R, big R. White ones are big R. The white ones are big W, big W, and the roan ones are big R, big W. They're a mix between the two, so they're actually going to be those two different colors. So to go over the first one, let's go over. It says what offspring are expected from a mating from a roan bull and a roan cow. So again, remember, bulls are males, cows are females. But these are two like RW, RW. So you would put RW here, RW here. You'd do your Punnett square, and then you'd see that you're going to get 25% that are red, 50% that are the roan, and 25% that are the white. So these ones, again, they do have a different heterozygous or a different like big R, big W phenotype. They're going to be the mix. No, they're going to be both red and white, not one of those colors. And there's a whole page for this as well. Blood typing. This is when you have multiple alleles that are going to be responsible for like blood typing. You have A, you have O, and then you have B as well. Remember that A and B are both dominant to O, and A and B are equally codominant to each other. So it asks you to put the genotypes for the blood types here. I typically allow students to use like the A's and the O's here, so A, B, and O, but I kind of try to say, hey, make sure you put the capitals for A, capitals for B, and lowercase for O. It's kind of hard to tell with those lowercase O's because they look like the bigger case O's, but hey, try your best. So like for homozygous A blood, this is going to be big A, big A. Heterozygous, remember, it's going to be big A, O. Not big A, little a, because that's not what we're talking about here. This is big A, O. Remember that the A's are dominant to the O's. Type O blood is just going to be OO. Same for the rest. You just got to figure out what these are. For the RH factor, remember, you can be positive, that's homozygous, positive, negative, that's heterozygous, and then the homozygous negative for RH factor would be negative, negative. And all you got to do is you got to cross these. You got to cross these like individuals in the Punnett square and tell me what the ratio of the blood typing is. So this one's homozygous for B, so it'd be big B, big B. This would be O, so she'd be OO over here. So they all have B blood, but they would be all heterozygous for B blood. So it'd be BO, 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 BO. And again, you have some of these questions here as well. But you can see here, this is a woman and a man, both heterozygous for the RH factor. So they would be positive, negative, positive, negative. So this would be positive, 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 negative, positive, negative, and then this would be negative, negative. So it would be 75% chance of having positive blood and a 25% chance of having negative blood. Same for these. It's going to ask you, again, more complex question here for number 17, but you can work through it. Sex-linked genetics. These work on the X and the Y chromosome, And all we have to do is we have to show whether it's a dominant or recessive trait on that X chromosome. Remember, males usually inherit these types of disorders or just genes more often because we only have one copy. So if we get one, we have it. It asks you to write down what the unaffected versus the carrier versus a female with hemophilia, and then an unaffected and affected male with hemophilia. Hemophilia is a sex-linked gene disorder. And so it gives you a bunch of these that are like, hey, cross these two. So we have the female, which is a carrier because she only has one of these genes, not both. A quote-unquote normal male. They have the big H here. So I'd put X big H up here with the Y, X big H, X little h here, and then do your cross. And it asks you what are the percent of all of them having hemophilia, and then the males versus the females as well. Remember, males are XY, females are XX, and it gives you different combinations and asks you, hey, what are these, or what are the outcomes between these two individuals? So this is my non-Mendelian genetics packet. You can work through it at your own pace. If you have a question, send me an email.